Okay, let's unpack the blueprint for life. Today, we're doing a deep dive into really the very beginning of human architecture.
That first structure.
Exactly. Tracking development right from that single fertilized cell all the way to building its own little life support system.
Yeah, the placenta.
Right. Our mission is to walk through the sources, focusing on that incredibly packed timeline, just the first 2 weeks.
It's kind of amazing, this sheer, let's say, organizational effort needed. Going from 2 cells to something ready to, well, implant itself.
And it starts, like you said, with fertilization, usually up in the fallopian tube, right? In the umbella.
Yeah, typical spot, yeah. And that location really dictates the early schedule. But before that fusion can even happen, the sperm has some prep work.
Right. It needs the right credentials, basically.
Yeah.
The sources mentioned 2 key things. First, capacitation.
Capacitation, yeah. It's a conditioning process that happens inside the female reproductive tract.
Okay. And then the acrosome reaction.
Exactly. That releases enzymes. Think of it like the key to unlock the egg's outer layer.
Yeah.
The zona pellucida.
A tough shell.
A very tough shell. And it's all precisely controlled.
Yeah.
The instant one sperm gets through, the egg throws up its defenses.
Like a biological firewall.
You could say that, yeah. A chemical reaction fires off immediately to block all other sperm. It's called preventing polyspermy, that single fusion. That's your zygote.
Okay, zygote formed, and then things get really interesting really fast. It starts dividing, right? Cleavage.
Cleavage. It's a rapid series of mitotic divisions, but here's the key thing. The overall size doesn't really increase much.
So it's dividing, but staying compact.
Exactly. From 2 cells, the blastomeres, to maybe 12, 16 cells by day 3. That little solid ball is called the morula.
And staying compact makes sense because it's still traveling down that narrow tube.
Precisely. It has to fit. Once it gets into the uterus, though, fluid starts to seep in,
Yeah.
and it changes shape.
Right. The solid morula becomes the holoblastocyst.
Yeah. And this is a huge moment. The cells sort of self-organize. They split into 2 distinct groups with different jobs.
The first big strategic move. You get the inner cell mass.
That's the future embryo proper.
And the outer layer, the trophoblast, that's the construction crew for the placenta.
Absolutely. And this construction, this bridge building, it kicks off really quickly. Implantation happens around day 6 after fertilization.
6 days.
And the placenta itself starts
Yeah.
forming just after that, maybe days 7 to 10. And it's a joint venture, remember?
Oh, a maternal.
At a maternal organ, yes. The fetus brings the chorion frondosum, derived from that trophoblast, and the mother contributes part of the uterine lining, the decidua bassalis.
And that trophoblast layer you mentioned, it's the powerhouse here.
Oh, absolutely. It immediately splits into 2 layers itself, an inner cytotrophoblast and an outer syncytotrophoblast.
Syncytotrophoblast.
Yeah, it's a mouthful.
Yeah.
But that outer layer, the syncytotrophoblast, is incredibly invasive. It pushes into the uterine wall, secreting enzymes, and crucially, it starts pumping out HCG.
Human chorionic gonadotropin. That's the hormone pregnancy tests look for.
That's one. It maintains the pregnancy early on. This invasion also drives the formation of these finger-like projections, the chorionic villi.
To increase surface area, I assume.
Massively increase it, yeah. And inside these villi, you get mesenchymal cells tuning into tiny capillaries, which then fuse to create this intricate network.
The arteriocapillary network.
And that whole structure, the placenta, becomes the fetus's complete transport system. Nutrition in, waste out, gas exchange, plus it manages hormones.
It's an incredible piece of biological engineering built so fast.
It really is.
Yeah.
Which brings us to the clinical side. We've seen this tight schedule, this choreography. What happens if things go off script?
Well, the risks seem pretty serious. If that blastocyst implants somewhere other than the uterus, like the fallopian tube.
Ectopic pregnancy.
Yeah, an ectopic pregnancy. The sources are clear. That's a medical emergency. Big risk of rupture, internal bleeding. Very dangerous.
Absolutely time critical. And even if it implants in the right place, there can be issues with where exactly or how deeply it attaches later on.
Like placenta previa.
Right. If the placenta ends up low down, covering the cervix partly or completely, that can cause serious bleeding, especially later in pregnancy or during delivery.
And the other one mentioned is placenta accreta. That sounds even more worrying.
It is. That's when those villus burrows are too deep into the uterine wall, almost fuse with it.
Making it hard to detach after birth.
Extremely hard, sometimes impossible without intervention. It can lead to catastrophic hemorrhage. So understanding these precise steps fertilization, cleavage, implantation, how those cells differentiate.
Right.
It's not just academic.
No, definitely not. It directly informs things like IVF, in vitro fertilization. Clinicians are basically replicating these initial stages outside the body.
Precisely. It's applied developmental biology.
So we've traced this amazing journey, this rapid construction project, building life's essential transport bridge.
We have. And considering that, here's something to think about maybe for you, the listener, as you wrap up this deep dive. We talked about the placenta as a transport system, but also a barrier. The sources note it's actually relatively thick in the first trimester.
Thicker early on, okay.
Yeah, when all those crucial organs are just beginning to form. So what might that initial thickness, that sort of physical distance, imply about the balance? The balance between vulnerability and protection right at the very start. Something to mull over. Welcome to the deep dive. Today, we're really getting into the weeds of early development. How does a simple ball of cells become, well, us?
It's an incredible process. We're looking at how that initial blueprint gets laid down, right? The first few weeks are just fundamentally important.
Absolutely. So our mission today is to unpack how those first crucial layers form, the germ layers, and also the support systems that, keep everything going early on.
Yeah, it all starts incredibly small. Around day 8, you've got the inner cell mass, the embryoblast, sorting itself out.
Into that flat two-layered thing, the bilaminar disc.
Exactly, the bilaminar embryonic disc. You have the epiblast on the outside, sort of, and the hypoblast underneath. Simple structure, really.
But simple doesn't last long. Week 3 hits and boom, gastrulation. That seems to be the game changer.
Oh, completely. Gastrulation is where you go from that flat disc to something with actual dimensions, depth, a plan.
And the key player there is the primitive streak.
That's the one. It appears on the back, the dorsal surface of that epiblast layer, and its appearance is profound.
Profound how?
Well, the moment it forms, it defines everything. The head end, the tail end, cranial, caudal, and crucially, left and right. It gives the whole embryo orientation.
Wow. Okay, so this streak directs traffic, essentially. Cells start moving.
Precisely. Epiblast cells start migrating inwards, diving down through the streak. It's quite the invasion.
And the first wave does what?
The first cells to move in actually push aside and replace the hypoblast completely. They form the definitive endoderm.
Endoderm. That's the innermost layer. What does it become functionally?
Think internal linings. So the lining of your entire gut tube, your respiratory system, that's all endoderm origin.
Okay. And then more cells follow.
Yep. The next wave slots in between the epiblast on top and the new endoderm below. This middle layer is the mesoderm.
Mesoderm sounds important. Muscle, bone.
You got it. All the structural stuff. Muscle, bone, cartilage, plus the entire cardiovascular system, heart, blood vessels, and the urogenital system too.
Right.
It's the body's framework and plumbing.
So endoderm inside, mesoderm in the middle.
What about the cells that didn't migrate? The ones left on top?
Those remaining epiblast cells, they now become the ectoderm, the outer layer.
Ectoderm gives us
skin. But also, critically, the entire nervous system, brain, spinal cord, and sensory organs. So interface with the outside world and the command center.
Incredible. 3 distinct layers, each with a massive job list, all from that initial migration through the streak.
It's the fundamental body plan, laid down in just days.
Now, you mentioned the primitive streak is temporary. What happens if bits of it linger?
Yeah, that can happen. Since the streak is the source of all 3 layers, if remnants persist, usually near the tail end, they can form a tumor called a sacrococcygeal teratoma.
And that's notable because?
Because it contains tissues from all 3 germ layers. You might find teeth, hair, ectoderm, muscle, bone, mesoderm, and gut-like tissue, endoderm, all in one tumor. It's a direct, albeit unwanted, consequence of that early developmental power.
Wow. And you also mentioned left-right asymmetry being set up then? Exactly. Defects in the genes controlling that process during gastrulation can lead to cetus in, versus where your internal organs are flipped, a mirror image, heart on the right, liver on the left.
Okay, let's shift gears slightly. While this layering is happening, support systems are forming too, right? Like the amniotic cavity.
Absolutely essential. This cavity forms sort of within the epiblast initially and gets roofed by cells called amnioblasts. It fills with fluid.
Amniotic fluid, the main job there is protection.
Protection is key, yeah. It's a shock absorber. But it also gives the embryo room to grow, to move, which is vital for normal limb development, for instance, keeps things from sticking together.
And it's the fluid they sample in amniocentesis later on.
That's the one. You can get fetal cells from it for genetic testing.
Okay, and below the disc, there's the yolk sac. Sounds important, but it's temporary.
Very temporary in humans, unlike in, say, birds. But its early roles are critical. First, it helps with nutrient transfer before the placenta is fully up and running.
Like a packed lunch.
Sort of. And crucially, it's the site of our very first blood cell formation. Hematopoiesis starts there.
But there's something else really vital it does, isn't there? Something about future generations.
Yes, this is amazing. The yolk sac is where the primordial germ cells originate. These are the precursors to sperm and eggs.
So the cells that will eventually form the gonads and determine reproductive capability start out in this temporary sac.
That's right. They are formed there, outside the embryo proper, and then they have to migrate into the developing body to find the gonads. It's quite a journey.
That's just astonishing. So within, 3 weeks or so, we've gone from 2 flat layers to a complex 3-layered structure with defined axes and the beginnings of all organ systems, plus these essential temporary support structures like the amnion and yolk sac.
The amount of coordinated activity and sheer functional output in that short window is, well, it's mind-boggling.
It really is. And that final point about the germ cells.
Yeah, think about that. The cells that hold the blueprint for the next generation arise in a structure that completely disappears, a temporary staging ground for genetic continuity. It's a beautiful piece of biological design, isn't it? Welcome back to the deep dive. Today we're looking into something pretty amazing, fetal circulation.
Yeah, it's a completely unique system, isn't it? Just for life inside the womb.
Totally specialized. It's designed really to bypass things that aren't quite ready yet, like the lungs obviously filled with fluid.
And the digestive system too, for the most part.
So our mission today, let's try and map this out fairly quickly. Understand these temporary shortcuts, these shunts that make it all work.
Exactly. Think of them like temporary roads, Only needed while the main highways, the lungs and stuff, are under construction. They disappear after the job's done.
Okay, let's start at the beginning. Where does the good stuff, the oxygen and nutrients, come from?
It all comes from the mother via the placenta, and it enters the fetus through just one vessel, the left umbilical vein.
Just one vein coming in, got it.
And right away, as soon as that blood hits the liver area, things get rerouted.
Already, okay.
Yeah. Most of that highly oxygenated blood, the best stuff, actually bypasses the liver tissue.
How does it do that?
Through something called the ductus venosus. It's like an express lane. It channels that blood straight into the inferior vena cava, the IVC.
Hold on, the IVC? But that's carrying deoxygenated blood back from the baby's lower body, right?
It is, yeah.
So we're mixing the really good oxygenated blood with, well, used blood right at the start. Seems a bit inefficient.
It sounds it, but it's strategic mixing. The resulting blend in the IVC is still pretty well oxygenated, much better than typical venous blood, and it sets things up for the heart.
Okay, so this mixed but still pretty good blood flows into the right atrium of the heart.
Correct.
Now, normally, in you or me, that blood goes right ventricle, then lungs. But the fetal lungs are offline, so what's bypass number 1 in the heart?
That would be the foramen ovale. It's basically a flap and opening between the right and left atria.
Ah, a hole between the chambers.
Exactly. And the pressure directs most of that incoming IVC blood straight through this opening, from the right atrium directly into the left atrium.
So it just skips the whole right ventricle and pulmonary circuit?
Largely, yes. It prioritizes getting that better oxygenated blood over to the left side of the heart, which pumps to the body, especially brain.
Why skip the right side so deliberately? What if it didn't?
Well, think about those lungs. They're collapsed, filled with fluid. The blood vessels inside them are constricted, creating huge resistance. That's like trying to pump water against a solid wall.
High pressure system.
Extremely high pressure. The right ventricle just isn't built to force large amounts of blood through that resistance. It would back everything up.
Makes sense. Okay, so most IVC blood goes through the foramen ovale. What about the blood coming back from the baby's upper body and head through the superior vena cava, the SVC? that also lands in the right atrium.
Right. And that blood does tend to go the normal route, sort of. It flows into the right ventricle.
And then the right ventricle pumps it towards the lungs.
It pumps it into the pulmonary trunk, yeah. But again, it immediately hits that high resistance from the lungs. Only a tiny bit actually goes into the lung tissue itself.
So where does the rest of it go? Is there another escape route?
There is. The second major cardiac bypass, the ductus arteriosus. This is a blood vessel connecting the pulmonary trunk directly to the aorta, specifically the arch of the aorta.
Ah, okay. So blood pumped towards the lungs gets immediately shunted over to the main body artery, the aorta, bypassing the lungs almost completely.
Precisely. The vast majority of blood leaving the right ventricle takes this shortcut into the aorta.
And this means the blood going up the aorta towards the head and arms gets the best possible oxygen mix available.
That's the key insight, yeah. The body prioritizes the brain and upper limbs with the most oxygenated blood it can manage through this shunting system.
Clever design. Okay, so blood circulates, delivers oxygen. How does it get back to the placenta to get refreshed?
Good question. As the blood travels down the descending aorta, some of it is directed into two umbilical arteries.
Two arteries going out, one vein coming in.
You got it. And these umbilical arteries branch off the internal iliac arteries. They carry the deoxygenated blood and waste products back out to the placenta for exchange with the mother system.
Okay, so we have these three key shunts. Ductus venous is by the liver, foramen ovale in the heart, ductus arteriosus connecting pulmonary artery and aorta. They're temporary, right? What happens if they don't close after birth?
That can cause problems.
Yeah.
It's often called persistent fetal circulation. Those specific issues have names. The most common one people might hear about is a patent foramen ovale, or PFO.
Patent meaning open, so the flap doesn't seal.
Exactly. Normally, when the baby takes its first breath, the lungs inflate, resistance plummets. Pressure on the left side of the heart suddenly becomes much higher than the right. That pressure difference slams that foramen ovale flap shut.
Like closing a door.
Pretty much.
Yeah.
But if it doesn't seal completely, you have a PFO. Often, it causes no symptoms at all. But the danger is that if a blood clot forms in the venous system, like in the legs, it could potentially travel up, slip through that PFO opening.
And go straight into the arterial circulation instead of being filtered by the lungs.
Yes. And if it goes to the brain, that can cause a stroke. That's the main risk with the PFO, even if someone feels fine.
Wow. Okay, that really highlights how crucial the transition at birth is. So, summarizing, the whole fetal setup relies on these temporary bypasses. Ductus venosus, foramen ovale, ductus arteriosus.
Absolutely. They define fetal circulation. And thinking about how the body prioritizes sending the best blood to the brain and upper body before birth, well, it leads to a fascinating question for you to think about.
Go on.
What precisely has to happen, structurally or maybe even chemically, in those first few moments, that first breath after birth, that completely flips the script? How does the system instantly switch priorities so the lungs and the adult heart pattern become dominant, overriding this incredibly complex fetal pattern that worked perfectly just seconds before? Right, let's dive into basically a construction project inside the embryo, building the gastrointestinal tract. Our focus today is how that whole system develops, starting right from the get-go with the primitive gut tube. Week 4 endoderm stuff.
Yeah, and it's surprisingly physical right at the start. You've got this folding happening, the cephalocaudal and lateral folding.
Okay, so folding turns a flat sheet into a tube.
Exactly. That's the first step. And once you have that basic tube, it immediately gets divided. Think like zones.
Zones.
Right.
The classic foregut, midgut, hindgut.
That's them. Each one has its own manufacturing list, you could say.
So foregut first. What's on its list?
Okay. Foregut is the top section. So you get the esophagus, the stomach, and the very first part of the small intestine, the proximal duodenum.
Ah, the duodenum. And isn't that where other things connect?
Precisely. The buds for the liver and pancreas, they actually sprout off the foregut right there.
That makes sense. Their ducts connect right around that area later on. Okay, so that's foregut. Midgut.
Midgut takes over for the bulk of the journey. Most of the small intestine, the cecum, appendix, and then the colon up to about 2/3 across the transverse part.
Wow, okay, that's a lot of territory.
It really is.
And hindgut finishes it off.
Yep. The last bit of the transverse colon, descending colon, rectum, and the upper part of the anal canal. The end of the line, basically.
So it sounds fairly linear when you list it like that, but things get twisty.
Yeah,
right,
like-
literally.
Oh, absolutely. The mechanics are fascinating. Take the stomach. It starts as just this sort of sausage-shaped bulge in the foregut,
Simple enough.
but then it rotates. It spins 90 degrees clockwise along its length.
90 degrees clockwise. Why?
That rotation is what gives it the final J shape. It pulls the greater curvature over to the left side, essential for how it functions, how it churns food.
Okay, a 90-degree twist for the stomach. But the midgut's journey is even more dramatic.
Oh, it's epic. The midgut grows so fast, elongates so much, that during the 6th week, it runs out of space inside the tiny abdominal cavity.
Runs out of space. So what happens?
It has to herniate. It pushes out, forms this U-shaped loop right into the base of the umbilical cord.
Whoa, wait. It actually pushes outside the main body cavity?
Yeah, into the umbilical cord. It's kind of mind-blowing. The liver and kidneys are also booming in size then, so they take up all the room. The gut gets, well, squeezed out temporarily.
Temporarily, so it has to come back in.
Right. And the return trip is the main event, really. Around the 10th week, as the abdomen gets bigger, the midgut loop starts to retract.
And it doesn't just slide back in.
Nope. As it comes back, it performs this incredible rotation, 270 degrees counterclockwise.
270 degrees, counterclockwise this time. That's a huge rotation.
It is, and what's really neat is how it does it. It pivots around the superior mesenteric artery.
They're the main blood vessel for that area.
Exactly. The artery acts like an anchor point, and the gut sort of spools itself neatly back into the abdomen around it.
That's some serious biological engineering. Okay, so we have tubes, rotations. What about other organs nearby? I remember reading something about the spleen being odd.
Ah, yes, the spleen surprise. It's not digestive at all, right? It's lymphatic, part of the immune system.
Right.
But developmentally, it actually forms within the dorsal mesentery that supports the stomach. So it ends up nestled right there with the foregut derivatives.
Strange neighbor.
Yeah.
So development isn't always strictly compartmentalized.
Definitely not. And, thinking about all this precise folding and rotating, it highlights how crucial timing and mechanics are if things go wrong.
Right, the clinical implications. Let's touch on those. Omphalocele, that sounds directly related to the midgut journey.
It is. That's basically when the midgut loop fails to return. It doesn't complete that retraction by week 10. So the baby is born with intestines, sometimes liver too, outside the abdomen, just covered by a sac.
A direct mechanical failure.
Exactly. Now contrast that with something like Hirschsprung's disease.
Okay, what's the issue there?
Hirschsprung's is different. It's not about large-scale movement. It's about something tiny, neural crest cells.
Neural crest cells, what do they do?
They're supposed to migrate all the way down the gut tube during development to form the nerve network, the ganglia, within the bowel wall. These nerves are essential for muscle contractions, for peristalsis.
So what happens in Hirschsprung's?
The migration fails. Those cells don't reach the final segment of the colon, usually the rectum or sigmoid, so that part of the bowel has no nerves.
And no nerves means?
It can't relax, it stays constricted, causing a blockage, severe constipation, abdominal swelling. It's a major functional problem, even though the structure looks mostly normal from the outside.
So we've got this incredible development story, massive physical movements like folding and that huge 270 degree rotation, but also these microscopic cellular migrations being just as vital.
It's a beautiful system, really. You need the big architectural changes, the twists and turns, but you also need the fine-tuning, the cellular components like nerves arriving correctly, even unrelated neighbors like the spleen settling in the right spot.
It really underscores the complexity. Failure can happen at the macro level, like the gut not retracting, or at the micro level, like cells not migrating.
And the functional outcome can be just as severe either way.
Which leaves us with a final thought for you, the listener. Think about that duality. How can a massive physical error, like organs staying outside the body, have a functionally similar catastrophic impact as the failure of tiny, unseen cells to reach their destination?
Yeah, what is this reliance on both sort of brute force mechanics and delicate cellular choreography tell us about how complex systems, maybe even beyond biology, are built and maintained? Something to chew on.
Welcome to the Deep Dive. Today we're looking at something incredible, how a tiny, almost balletic twist deep inside us, weeks after conception, sets up a vital organ. We're talking about the pancreas. and specifically how its development can sometimes lead to problems later in life, like pancreatitis risk.
It's a fascinating journey, really. It all kicks off around the 5th week of gestation. The pancreas doesn't just appear fully formed. It actually starts as 2 distinct pieces, little buds growing out from the early gut tube, the duodenum.
2 buds. Okay, so how do they, divvy up the job of building the final pancreas?
Well, there's a bigger one, the dorsal pancreatic bud. That one's higher up. It takes on the lion's share, forming the main body, the tail, and the upper part of the pancreas head.
And the second one, the smaller one?
That's the ventral pancreatic bud. It's smaller, yeah, but its position is key. It develops down near the bile duct. It's destined to form the lower head section and that little hooked part called the uncinate process.
Right, the uncinate. So you have these 2 separate pieces. How do they become one organ? This is where that rotation comes in, isn't it, around week 7?
Exactly. This is the crucial move. As the duodenum itself grows and rotates into that C shape we recognize in the abdomen, it literally pulls the ventral bud along with it, drags it around towards the back.
Ah, so it swings behind.
It swings dorsally, yeah, until it lines up right next to the dorsal bud. And then they fuse. That merger creates the single pancreas we know.
And the plumbing inside has to merge too, right? The ducts carrying the digestive juices.
Absolutely critical. The main pipeline, the main pancreatic duct, or duct of Wirsing, forms mostly from the ventral buds duct. But it also incorporates the tail end, the distal part of the dorsal buds duct.
Okay, so what happens to the beginning part of that dorsal duct?
Usually that proximal part just kind of fades away, or it might stick around as a smaller secondary channel. That's the accessory duct, the duct of Santorini.
Got it. And while all this structural stuff is happening, the hormone factories are setting up shop too. The islets of Langerhans.
Precisely. Those endocrine cells making insulin and glucagon, they develop within the tissue and spread throughout this newly fused gland. It's all happening simultaneously.
Such a complex sequence. So it makes sense that if something goes wrong with that rotation or fusion, there can be consequences.
Definitely. These developmental hiccups are behind several congenital anomalies.
What's a major one? When does it usually cause problems?
Well, one of the really dramatic, though thankfully quite rare, conditions is annular pancreas. Here, the ventral bud does something strange. It seems to split, and parts of it migrate in opposite directions around the duodenum.
Opposite directions, so it encircles the gut tube.
Exactly. It forms a complete ring, an annulus of pancreatic tissue squeezing the duodenum.
Wow. And that obviously causes a blockage.
A major one. You might see it present very early, like in newborns with vomiting, feeding problems, sometimes even excess amniotic fluid before birth because they can't swallow it properly. In adults, it might be less obvious initially, but can cause ongoing issues like pain or, yes, recurrent pancreatitis due to that constriction.
Okay, that's the rare severe one. What about the most common issue? I think you mentioned pancreas divisum.
Pancreas divisum, yeah. By far the most common anomaly. This happens when that final fusion step between the 2 duct systems just fails. They stay separate.
So the plumbing remains divided. What does that mean functionally?
It means most of the pancreas, the bulk of it, the body and tail derived from that big dorsal bud, has to drain all its enzymes out through that tiny little accessory duct. Remember, that accessory duct usually shrinks or disappears. Here, it's the main outlet for the largest part of the gland, draining through the minor papilla.
That sounds inefficient, like a bottleneck.
It can be. For many people, maybe most, it causes no symptoms at all. They live their whole lives unaware. But for some individuals, that potentially inadequate drainage pathway seems to be a risk factor. It might predispose them to recurrent episodes of acute pancreatitis. The drainage just can't keep up sometimes.
Interesting. And quickly, what about bits of pancreas ending up in totally the wrong place?
Ah, yes, ectopic pancreatic tissue. That's also seen sometimes. Small patches, little islands of pancreatic tissue just found elsewhere in the GI tract. Most often maybe in the stomach wall or the duodenum, even in a Meckel's diverticulum. Usually harmless, totally asymptomatic.
But I guess, like any pancreatic tissue, it could potentially get inflamed.
It could, yes. Though it's not common for it to cause problems, still it's another example of how complex this development is.
So wrapping this up, it's really striking that the fundamental structure, the very function of an organ, as critical as the pancreas, hinges on this intricate rotation and fusion happening way back in the 7th week of development.
It really puts things in perspective, and it leads to a final thought, maybe. Given that pancreas divisum, the most common variation, is often silent, it makes you wonder, doesn't it? What other subtle, maybe tiny anatomical variations might be present in any of us right now? Variations that don't cause issues day to day, but could potentially influence our health or resilience only under very specific circumstances, like illness or significant stress. Something to ponder.
Okay, so let's get into this. So we're looking at how the heart developed from just a simple tube, right?
Yeah, exactly. It's pretty incredible. We're talking about the first major organ getting up and running, starting way back in like the third week.
Third week, wow. And it goes from a tube to this complex 4-chamber thing.
That's the journey. It starts with 2 endocardial tubes fusing, makes one primitive heart tube.
And this tube isn't just blank, is it? It already has parts.
Right. It's already zoned, you could say. You've got the truncus arteriosus, bulbus cordis, primitive ventricle, primitive atrium, and the sinus venusus. 5 distinct regions already sort of programmed.
Okay, program.
Yeah.
But before dividing anything up, it has to move.
Yeah.
This cardiac looping.
Oh, yeah. Looping is absolutely crucial. Before any walls go up, the whole tube has to bend and fold.
Why? What does that actually do?
It physically positions the future chambers. It needs to loop to the right, that's called delooping, to get the ventricles and atria basically where they belong anatomically.
And if it doesn't loop right?
Yeah.
If it goes left?
Well, that's where you can get dextrocardia. The heart ends up on the right side of the chest. It shows how critical that very first directional fold is, you know. One wrong turn early on changes everything.
Huh. So placement first, then the internal walls start forming. What about that receiving area, the sinus venusus?
Right, so the sinus venosus takes in all the venous blood initially, and its fate is kind of interesting.
How so?
Well, the right horn doesn't just vanish, it actually gets absorbed, like fully integrated into the developing right atrium. It forms that smooth-walled part we call the sinus venerum.
Okay, so it becomes part of the atrium.
Exactly. And the left horn, it mostly shrinks down to become the coronary sinus.
Which drains the heart muscle itself.
Yep, so that primitive collector gets repurposed.
All right, now the big divisions. Let's talk about the ventricles. The interventricular septum, or IVS, starts in week 4.
Yeah, around week 4, and this is where things get really complex structurally speaking. It's got 2 main parts.
The muscular part and the membranous part.
Correct. You've got this thick, crescent-shaped muscular bit growing up from the bottom of the ventricle. That part's usually pretty solid.
Okay.
But it doesn't reach all the way to the top initially. There's a gap.
And that gap is closed by the membranous septum.
Yes, and that's the tricky part. It's much thinner, more delicate.
Because it's not just one thing growing, right? It's a meeting point.
Precisely. It's a fusion zone. You need 3 different structures to come together perfectly.
Okay, what is...
You need the top edge of that muscular IVS we just mentioned. You need tissue from the endocardial cushions kind of growing down. And critically, you need a contribution from the aorticopulmonary septum.
The aorticopulmonary septum. Okay, hold that thought. So these 3 things fuse.
They have to meet and fuse seamlessly.
And if they don't?
That's your ventricular septal defect, a VSD. The most common congenital heart defect, actually. Usually it's that membranous part that failed to form or fuse correctly, a hole between the ventricles.
Wow, okay. So that aorticopulmonary septum you mentioned, involved in closing the VSD gap, it does something else too.
Yes, huge other job. It's responsible for dividing the main outflow tract, the truncus arteriosus,
Dividing it into
the 2 great arteries, the aorta and the pulmonary trunk. It forms from these things called bulbar ridges.
And crucially, it doesn't just divide it straight down, does it?
No, and this is key. It has to spiral. It forms a spiral septum. That twist is essential.
Why spiral? What happens if it just goes straight?
If it fails to spiral, if it divides straight, you get a catastrophic defect called transposition of the great arteries, or TGA.
Transposition.
Yes.
So the arteries are switched.
Exactly. The aorta incorrectly comes off the right ventricle, and the pulmonary artery comes off the left ventricle.
Which means, the oxygenated blood just goes back to the lungs, and the deoxygenated blood just keeps going to the body.
Right. 2 separate circuits that don't mix properly. It's lethal without immediate intervention. That spiral is non-negotiable for a functioning circulation.
Okay, so we've covered ventricular separation and the outflow tract division. What about the atria? Holes there are also common, right? ASDs.
Absolutely. Atrial septal defects, ASDs. Different mechanism, but also involves incomplete partitioning.
So how does that division work? Is it also just one wall?
No, it's more like 2 overlapping curtains. You have the septum primum growing down and then the septum secundum growing next to it.
And they leave a hole. Intentionally.
Well, before birth, yes, they form a flap valve called the foramen ovale, which is crucial for fetal circulation. But after birth, it's supposed to seal shut.
So an ASD happens if those curtains, the septum primum or secundum, don't grow right or don't overlap properly.
Exactly. If there's not enough tissue or if the overlap fails, you're left with a persistent hole between the atria after birth.
Man, when you lay it all out, the initial looping, the fusion for the IVS, the spiraling for the great arteries, the overlapping septa in the atria.
Yeah.
It's just layer upon layer of these precise events that have to happen perfectly in sequence in that first month or so.
It's an incredible architectural feat, really. The precision required is mind-boggling.
It makes you really think, doesn't it? How many things have to go exactly right just to get that basic four-chamber structure functioning?
Yeah.
It really highlights the fragility and the wonder of it all.
Welcome to the deep dive. Today, we're zeroing in on a really intricate bit of biological architecture. We're talking about how the female internal genitalia actually developed from the very beginning, in the embryo. Our goal here is to get a quick handle on the steps involved, and maybe more importantly, what happens when things go, well, a bit wrong structurally.
It's a fascinating area, and what's really striking is that this whole complex structure arises mostly because certain signals are missing. It's almost like a default setting.
A default? How so?
Well, it all comes down to two sets of ducts you start with. The mesonephric ducts, also called Wolffian, and the paramesonephric or Müllerian ducts.
Yeah.
In female development, the key is the absence of testosterone and also the absence of something called anti-Müllerian hormone, or AMH.
Okay, so let's start there. No testosterone. What happens to those Wolffian ducts, the ones that would normally form male structures?
They basically just regress. They degenerate, get out of the picture entirely because there's no testosterone signal to maintain them. Okay, they're bypassed. So the Müllerian ducts then take center stage because there's no AMH.
Exactly.
No AMH means the Müllerian ducts stick around and they become the foundational blueprint for the female internal tract.
Right. So how do these Müllerian ducts actually build the system? What forms what?
Okay, so think of these two parallel tubes. The top parts, the cranial ends, they stay separate. These unfused bits develop into the fallopian tubes, with the little finger-like ends called fimbriae.
Okay, the tubes. Makes sense. But the uterus and vagina?
That's where the critical next step happens. The lower portions, the caudal ends of these two Müllerian ducts, they have to grow towards the middle, towards each other, and then they fuse. Like zipping up. This fused structure is what we call the uterovaginal primordium.
The uterovaginal primordium. Got it. And that gives rise to...
That forms the uterus itself and also the upper sort of 1/3 of the vagina.
Well, wait, you said they fuse. Isn't there like a wall between them initially?
Yes, absolutely. Good point. After they fuse side by side, there's still a central wall, a septum, right down the middle. And this septum must break down. It has to resorb completely to create a single open uterine cavity. That resorption step is crucial.
Okay, fusion, then resorption. That clarifies things. So the uterus and the top of the vagina come from these fused Müllerian ducts. But you mentioned earlier the lower 2/3 of the vagina form differently.
That's right. It's a sort of bottom-up process for the lower part. Where the fused Müllerian ducts touch the urogenital sinus, another embryonic structure, they trigger the formation of something called synovaginal bulbs.
Synovaginal bulbs.
Yeah, think of them like little outgrowths. These bulbs proliferate and form a solid plate of tissue that grows upward. Then, this solid plate has to undergo canalization. It needs to hollow out to form the lower 2/3 of the vaginal canal and connect with the upper part.
Wow. So many steps needing perfect timing. Fusion, resorption, canalization.
Precisely. And you can see how, with this level of complexity, errors can happen. Which leads us directly into the clinical side.
Right, the anomalies. Where do things typically go wrong?
Well, you can broadly group them. Problems with fusion and problems with that resorption step we talked about.
Okay, fusion failures first.
If those lower Müllerian ducts completely fail to fuse, you get what's called uterus didelphys. Essentially two separate uterine bodies, often two cervices, sometimes even two vaginas.
A complete duplication. And if it's only partial failure,
that leads to a bicornuate uterus. It fuses lower down, but not fully at the top, creating a kind of heart shape with two distinct horns.
Okay. But you mentioned resorption failure too. That's different.
Yes. And this is actually the most common type of anomaly,
Yeah.
the septate uterus.
And this is the one I've read is really significant for recurrent pregnancy loss.
Exactly that. Here, the fusion happened correctly, the ducts met and joined, but that central wall, the septum, didn't resorb properly. It leaves a partition inside the uterine cavity, which can interfere with implantation or fetal development.
That makes sense why it's so clinically relevant. Okay, what about the last step? The hollowing out of the lower vagina. Canalization failure.
If that sinovaginal plate doesn't canalize, doesn't hollow out, you get vaginal atresia. The lower part of the vagina is essentially closed off or might be missing altogether.
And how would that typically present?
Usually at puberty. A young woman will start developing secondary sexual characteristics, but she won't have periods. That's primary amenorrhea. But she might have cyclical pelvic pain because the menstrual blood is being produced but can't get out. It backs up into the uterus and tubes, a condition called hematocolpos.
Trapped menstruation. Okay, one last thing. What about those Wolffian ducts that were supposed to disappear? Do they ever leave traces?
They can. Sometimes small remnants persist. These can form fluid-filled cysts along the path where the duct used to be, usually in the vaginal wall. We call them Gartner cysts.
Gartner cysts, like leftover plumbing.
That's a perfect way to put it. They're usually harmless, often found by chance during an exam, but they're a neat little reminder of that initial dual duct system.
And so we've traced this incredible journey from two simple pairs of ducts through fusion, resorption, canalization, all leading to the final structure. The timing really is everything.
It truly is. And maybe the final thought to leave you with is just how profound that default mechanism is. Think about it. The fundamental difference determining whether an embryo develops this entire female internal system or the male internal system boils down simply to the absence of two hormones. The body has this inherent plan just waiting. And if those specific male determining signals don't arrive, this intricate female structure is what unfolds. It's quite remarkable when you consider the power of that absence.
All right, let's get into it. Today, we're really looking at the foundational stuff, the pharyngeal arches.
Yeah, the absolute blueprint.
And how they basically dictate whether you end up with, a normal tongue or potentially some serious issues with your arteries. It's all laid down right at the start.
Exactly. And our mission for this deep dive really is to pull out the key bits from the sources, looking at how these initial segments become things like the tongue and those major arteries feeding the brain and lungs.
It's fascinating. It really is like an architectural plan.
It is, yeah. A very intricate one.
So the tongue seems like a good place to start. It's not just one single muscle popping up, right? It's more like a construction job.
That's a great way to put it. It involves very precise fusion.
So the front part, the anterior 2/3, that's what we use most for talking, right?
The whole section comes from the very first pharyngeal arch.
Yeah.
You've got these things called lingual swellings. And a central bit, the tuberculum impar, they merge together.
Okay, so first arch for the front. But the back is different.
Totally different origin. The posterior 1/3, that comes from the third and even the fourth arches.
Huh.
Yeah, through something called the hypobranchial eminence. It's like a whole separate module that has to, migrate and fuse perfectly.
And you can actually see where they meet.
You can. Even in adults, that V-shaped line, the terminal sulcus, that's the fusion point. Marks the boundary between those two different developmental zones.
And if that fusion doesn't happen just right, then problems arise.
Immediately. Yeah. Like if those lingual swellings just grow too much, you can get macroglossia.
An abnormally large tongue.
Right. Or something more common perhaps. Ankyloglossia.
Yeah.
Tongue tie. That's where the little bit underneath the frenulum is too short or tight.
Affects feeding, speech, all that.
Exactly. All down to how those initial bits developed and fused.
Okay, so that's fusion. But you mentioned regression too, things disappearing. Let's talk about the arteries, because that seems like a big part of the story.
Oh, absolutely. It's maybe even more dramatic with the arteries. What's really striking is how much of the original system is just temporary.
Temporary, like placeholder arteries.
Kinda, yeah. There were initially 6 pairs of these arch arteries. Most of them just regress. They pop up, maybe do something briefly, then vanish. But the ones that stick around, absolutely critical.
Okay, so which ones survive?
Well, the third arch artery is a huge player. It doesn't regress. It forms the common carotid artery.
The main highway to the head, basically.
Exactly. And the internal carotid, too. So, yeah, a massive contribution from just that one embryonic artery.
What about the others? You said 6 pairs.
Right. The fourth arch artery. Now, this is where it gets really interesting because it's asymmetrical.
How so? Different on the left and right.
Completely different fates. On the right side, it helps form the right subclavian artery, feeding the right arm.
Okay.
But on the left side, that same fourth arch artery becomes part of the actual arch of the aorta, the main vessel leaving the heart.
Wow. Why the difference? Is that common in development?
It is in this specific context, yeah. It's all driven by how the heart and the great vessels loop and position themselves during development. It sort of forces this asymmetry.
Fascinating. What about the 5th?
The 5th, it's basically a ghost. It appears super briefly, seems to do nothing important, and just regresses entirely. Gone.
Nature being efficient, maybe? Or just messy?
Maybe a bit of both.
Yeah.
But then you have the 6th arch, and this one is crucial, especially for the lungs.
Pulmonary system.
Exactly. It gives rise to the pulmonary arteries. And importantly, on the left side, the distal part of the 6th arch artery forms the ductus arteriosus.
Ah, the ductus arteriosus. I know that one. That's the fetal shortcut, right? Bypasses the lungs before birth.
Precisely. Vital in the womb. But it absolutely has to close after birth if it stays open.
That's patent ductus arteriosus. PDA.
That's it, PDA. You get a mixing of oxygenated and deoxygenated blood, which is not good.
And isn't there another issue right around there? Coarctation?
Yes. Coarctation of the aorta. It's a narrowing of the aorta,
Yeah.
often right near where the ductus arteriosus attaches.
Which restricts blood flow out to the body.
Severely. Makes the heart work way too hard. Big pressure problems. It really highlights how critical these developmental steps are.
So, putting it all together, complex things like our carotids, our tongue, they start as these separate little embryonic pieces.
Absolutely. Distinct segments that have to survive or regress or fuse, all on a very strict schedule. It's intricate timing.
Microscopic building and demolition work, basically.
That's a perfect analogy. So think about this. We talk about the 1st, 3rd, and 4th arches for the tongue, and the 3rd, 4th, and 6th for the main vessels.
Right.
We barely mentioned the 2nd arch, except that its artery mostly regressed. But it was there. What other structures, maybe things around the jaw or even the ear, must have depended on that 2nd arch during early development before it, faded back?
Good question. Something to think about.
Definitely something for you to explore further. What really struck me from our sources was just how intricate, how incredibly precise that early facial development has to be.
Oh, absolutely. Like a tiny biological ballet.
Exactly. So today we're diving deep into the embryology of the lip and palate. We really want to focus on what happens when that facial fusion goes wrong.
Right. Pinpoint the failures.
Yeah, nail down where the process breaks down and, what that actually means for the individual.
And it's such a compressed timeline. It's quite amazing. The whole critical period for the lip and palate forming and fusing is basically between the 4th and 10th weeks of gestation.
Just 6 weeks.
A disruption in that tiny window, the consequences are, significant and lifelong.
Okay, so let's try and unpack this construction project. Stage 1 seems to be about the upper lip and what's called the primary palate.
That's right.
You get the central part of the face, the medial nasal prominences, meeting the bits coming from the sides, the maxillary prominences, and when they join up, that forms the very front section of the hard palate.
Exactly right. But that primary palate, it's actually only a small part. Think of it like the front porch of the palate, just anterior to the incisive foramen.
Okay, so just a small piece.
Yeah, the main structure, the secondary palate, which is most of the hard palate, and all of the soft palate that forms differently.
Right, and this is where things get really interesting almost. Acrobatic, like you said, it forms from 2 shelves, growing from those side maxillary prominences.
Precisely. But initially they grow downwards,
Yeah.
vertically, sort of like curtains hanging down on either side of the developing tongue.
Okay, so they're hanging down. How do they fuse them?
They can't fuse like that. They actually have to elevate. It's quite dramatic. They swing upwards from vertical to horizontal.
Yeah.
Imagine them rotating up to meet in the midline, above the tongue. Then they fuse together.
And this all has to happen by
the 10th week. That's the deadline for complete fusion.
Okay, so now we can link specific problems back to these stages. The cleft lip, it maps back to stage 1, right?
Exactly. It's a failure of that maxillary prominence, the side part, to fuse properly with the medial nasal prominence, the central part.
And the severity can vary quite a bit.
Oh, definitely. It can be just a tiny notch in the lip, or it might be a complete separation going all the way up into the nostril. And it can be on one side unilateral or both bilateral.
Does a bilateral cleft automatically mean it's more severe in terms of impact? Or is it really about the depth of that separation?
Well, the depth is certainly critical for function and repair, but yeah, a bilateral cleft often suggests a more significant disruption in that whole initial fusion process. It definitely complicates things.
Got it. Now, the palate, cleft palate is different.
It is. Cleft palate is about the secondary palate. It's when those two shelves, those swinging doors we talked about, fail to fuse.
You with each other in the middle.
Right, or they might fail to fuse with the primary palate structure at the front.
And the result is?
An opening. A direct connection between the oral cavity, the mouth, and the nasal cavity above it.
So what does that mean immediately for a newborn? What are the big challenges?
Functionally, the immediate big one is feeding.
Because they can't create suction.
Exactly. You need a complete palate to seal around a nipple, whether breast or bottle, to draw milk effectively. Without that seal, it's very difficult.
Leading to issues with weight gain, I imagine.
Yes, poor weight gain is a common early concern. The other major issue comes a bit later: speech.
Right. You need the palate to form sounds properly.
Absolutely. Many sounds require you to direct air pressure either through the mouth or nose. With an opening in the palate, that control is lost, making articulation very difficult.
Which explains the surgical timing, doesn't it? Lip repair is often done quite early.
Yeah, typically within the first few months of life, the lip repair is often more straightforward, aesthetically important, and helps with initial feeding to some extent.
But the palate repair waits longer.
Usually, yes. Typically around 12 to 18 months, and that timing is deliberate.
Why then specifically?
It's time to coincide with the period when children are really starting to develop speech sounds. Repairing the palate just before that crucial language explosion gives them the best chance to develop normal speech patterns.
It's just incredible how these significant functional challenges trace directly back to specific moments being missed in that, 6-week window, weeks 4 to 10.
It really highlights the complexity. And when you think about the mechanics, these two large shelves needing to grow, then elevate, then move medially, then fuse perfectly, all by week 10.
So much have to go right.
Exactly. Which leads to a final thought, maybe something for you, the listener, to consider. Given this intricate, precisely timed dance, what does the failure tell us? What underlying triggers, genetic, environmental, maybe a mix, could disrupt such a complex, multifactorial process? That susceptibility is definitely something worth exploring further.
Welcome to the deep dive. Today, we're cracking open something pretty fundamental. Odontogenesis, basically how your teeth develop.
Yeah, and what's really striking is just how early it all begins. We're talking the 6th week.
6th week of embryonic development.
Yeah.
It just seems incredibly fast for something so well intricate.
It really is. And the mechanism itself, it's based on this dialogue, really. We call it reciprocal induction.
Okay.
So it's not just one set of instructions. It's a two-way conversation, a very precise one, between the oral epithelium and that underlying ectomesenchyme tissue, like building with two teams, constantly checking in.
A coordinated construction. I like that. So, okay, our mission for this deep dive. Let's quickly map out those stages from the very first sign to the finished tooth.
And then maybe look at what happens when things go a bit sideways, the clinical side.
Exactly. Those fascinating exceptions when the blueprint isn't followed perfectly.
Right. So the foundation gets laid down almost right away.
Yeah, it starts with the dental lamina stage. That's basically the oral ectoderm, the surface tissue just thickening up, forms like a U shape.
Where the jaws will eventually be, yeah. And then straight into the bud stage. That's where you see those first little knots forming.
10 of them, right. The beginnings of the baby teeth in each jaw.
Exactly. 10 primordia per jaw. Then things get more differentiated.
With the capstase.
Uh-huh. The ectomesenchyme part starts to change. It kind of folds in, and you get two key parts forming from it. The inside bit becomes the dental papilla.
And that eventually becomes the pulp, the living bit inside.
That's the one, the nerve and blood supply core. And then the tissue surrounding that is the dental sac.
The support crew, you called it.
Yeah, you could say that. Because that dental sac, that's crucial. It's going to form everything that holds the tooth, the cementum on the root, the periodontal ligament.
The shock absorber.
Right, and even the alveolar bone socket it sits in. So the whole anchoring system is being planned out here.
Okay, so now things really specialize in the Bell stage. This is where the specific jobs get handed out.
Precisely. It's all about cell differentiation. The cells lining the dental papilla, they become odontoblasts. Their job, making dentin. That's the main bulk of the tooth under the enamel.
And the enamel comes from different cells.
Yep. The inner enamel epithelium cells, they transform into ameloblasts, and they make the enamel. But, and this is key, dentinogenesis making dentin has to happen first.
Ah, so the odontoblasts lay down the dentin layer.
Kind of like the mold, yeah. It dictates that inner shape, and then the ameloblasts come along and lay down the enamel over it.
That super hard outer shell, the hardest stuff in our bodies.
Exactly, so the foundation dictates the armor, like you said.
Okay, so once that crown is built, this miniature masterpiece, then what? Root time.
Then it's root time. Root formation happens after the crown's done, guided by something called Hertwig's epithelial root sheath. And only after that does the tooth finally erupt.
Quite a journey. But as you mentioned, sometimes this intricate dance falters.
Right. And the clinical consequences can be pretty significant. Take amelogenesis imperfecta. If those ameloblasts don't do their job right during that Bell stage.
The enamel formation goes wrong.
Exactly. It's actually a group of hereditary conditions.
Yeah.
The enamel is weak, maybe discolored, thin. So the teeth are sensitive, they wear down fast, break easily.
Not good. And then there's the one that just sounds strange. Dens and Dante.
Yeah, the tooth within a tooth. It sounds bizarre, but it's a developmental anomaly. Basically, part of the outer surface folds inward before the tooth hardens.
Creates like a little pocket.
Pretty much, an invagination. And that spot is incredibly vulnerable because the enamel in that fold is often thin or, well, just not there. So bacteria get in easily.
Leading to quick decay and pulp infection.
Very quickly, yeah. It needs careful watching for treatment.
And we should briefly touch on natal teeth.
Ah, yes. Teeth someone's actually born with. Sometimes they're just early versions of normal baby teeth, maybe a bit underdeveloped.
But they can sometimes signal other issues, right? Underlying syndromes.
They can, yeah. So they always need checking out. Plus, if they're loose, they could be an aspiration risk, or they might interfere with feeding, causing irritation. Sometimes they need to be removed.
So even these tiny early structures can have immediate implications.
Definitely. A small flaw, big consequences sometimes.
So wrapping this up for you listening, we've seen this incredibly rapid construction project starting at just 6 weeks. Specialized cells like ameloblasts creating literally the hardest substance in the human body. It's just an amazing feat of biological timing.
Absolutely amazing. And it leaves you with a final thought, maybe. Remember that dental sac? It forms the cementum, the ligament, and the bone holding the tooth.
All the support structures.
Right. So how deeply interconnected are those supporting tissues with the tooth itself, throughout your entire life? Their health relies on each other, something to chew on next time you're thinking about your teeth.